Assalamu alaikum and very good afternoon to everyone. I warmly welcome all our distinguished researchers, members, and participants from across the globe to Day 1, Session 4 of 8th International Conference on Applied Zoology 2025, organized by Department of Zoology, University of Punjab, in collaboration with other prestigious institutions. I'm Aliza Aleem, and it's truly an honor to host this exciting session filled with insightful presentations. Our today's session focuses on physiology, wildlife, microbiology, and drug discovery, bringing together innovative studies that connect fundamental science with applied zoological research. Our presenters will share valuable contributions addressing everything from microbial potential, animal physiology, to wildlife health. So, without further delay, I would like to start our first presentation. So, for our first presentation, I would like to welcome Dr. Mahwish Salman from Government College University, Faisalabad, and her topic of presentation is Evaluation of Antibacterial and Mosquito Larvicidal Potential of Brachybacterium conglomeratum.
Bismillahirrahmanirrahim. First of all, I would like to thank the organizers of this conference, the Applied Zoological Society of Pakistan, in collaboration with University of Punjab, Lahore, and Government College University, Faisalabad. Thank you for giving me opportunity to present my research work. The title of my presentation is Evaluation of Antibacterial and Mosquito Larvicidal Potential of Brachybacterium conglomeratum. These are the outlines of my presentation. First of all, I would like to introduce my title, The Vector-Borne Diseases Affecting Animal and Human Health, Several Mosquito Species of Culex and Anopheles are pathogenic vectors of many diseases. Many synthetic insecticides, such as organophosphates, are used to control these biological vectors, but their use as an insecticide has been prohibited due to the risk issues. Of these alternatives, natural bioactive metabolites that exhibiting unusual structures isolated from microorganisms have been playing a significant role in drug discovery. Actinobacteria and their metabolites have potential bioactivities. That's why we have selected one of the actinobacteria, that is Brachybacterium conglomeratum, that has been reported as a naturally occurring microorganism and belongs to family Micrococci, class Actinobacteria, and order Actinomycetes. The aims and objectives of my research is the fermentation production of secondary metabolites from this brachybacterium, the extraction of its metabolites, and the identification of metabolites by GCMS, and to perform its mosquito larvicidal activity. Now comes to the methodology. First of all, we have to uh, optimize the growth conditions of Brachybacterium conglomeratum that was previously isolated from maize roots and cultivated on LB medium at 37 degrees Celsius. Our next task is to collect and rear the mosquito species that was collected from a specific region in Faisalabad after rainfall. The methodology is further moved to the fermentation production of secondary metabolites from the selected bacterium and the extraction of these metabolites and the identification of these metabolites, and then we have to perform the following activities. Now move to the results. After fermentation production, we have after centrifugation and harvesting, we have collected a micropore filtrate after passing through 0.45 milli pore filters, that was further used for extraction by ethyl acetate solvent, that is a solvent-based extraction, and we get the extract which has a lot of metabolites that was further used for to perform the bioactivities. And before that, we have first identified the bioactive metabolites that was um, found or detected by GCMS. That is a chromatogram, and these are the different compounds that the peaks representing that identification, the detection of different compounds in this bacterium. Then we move to antibacterial assay. The antibacterial assay was performed against different pathogenic bacteria. The plate A that shows the antibacterial activity against Staphylococcus aureus, B is against Escherichia coli, C is against Bacillus cereus, and D is against Serratia marcescens. And these are the inhibition zones in millimeter. You can see the brachybacterium that has the brachybacterium secondary metabolites have maximum bioactivity against Serratia marcescens. Next is the mosquito's larvicidal activity, which was performed against Culex cuncunfasciatus larvae. Uh, and this slide, it shows mortality means of this larvae at different time intervals and concentration of bioactive secondary metabolites. You can see there is a exposure time of 24 or 48 and 72 or and there is a concentration in ppm of extract of brachybacterium in 50 ppm, 100, 150, 200, and 250 ppm. And we can observe that we found maximum mortality after 72 hours at 250 ppm. Similarly, this is the LC50 value, and we found uh, the minimum LC50 value after 72 hours, that is 49.86. This one is the acetylcholine esterase activity, and we found this activity at 50 ppm, 100, 150, 200, and 250 ppm. And we concluded that at 250 ppm, we have maximum acetylcholine esterase activity, that is of 56.44%. Now, we have to conclude our presentation. The Brachybacterium conglomeratum that have maximum secondary metabolites, bioactive secondary metabolites that have been found effective against various pathogenic microorganisms. And no doubt, they also have potential against mosquito larvae of Culex fasciatus, and also modulates the acetylcholine esterase enzymatic activity that caused the death of these larvae bacteria. And it was concluded as that the use of bioactive secondary metabolites from the microorganisms, that will be a good alternative of synthetic insecticides. Thank you so much for your attention.
Thank you so much, Dr. Mahwish Salman, for antibacterial and mosquito larvicidal potential of microbial species. It was truly lightning. For our next presentation, I would like to invite Ms. Nawara Ahmed from UF, and her topic of presentation is evaluation of button mushroom feed on hematobiochemical and gut health of Labiorhita rahu. Overs to you, Ms. Nawara.
Bismillahirrahmanirrahim. I am very thankful for the 8th International Conference on Applied Zoology 2025 organized by the Applied Zoological Society of Pakistan for providing us the platform to present our research findings. I am Nivera Ahmed, ICAZ Presenter 2025 from Department of Zoology, Wildlife and Fishes, University of Agriculture, Faisalabad. Today I am going to present my research topic entitled Evaluation of Button Mushroom Feed on Hematobiochemical Parameter and Gut Health of Labiorrhea. Aquaculture is the fastest growing food sector, supplying over 50% of the global fish. As we know, synthetic feed additives like the antibiotics creates risk of antibiotic resistance and health hazards. Therefore, there is a need for sustainable and natural alternative to these antibiotics. White button mushroom was best fit for it because it contained natural elements like the beta-glucans, antioxidant, fibers, non-immunostimulant. There was no previous study was done on white button mushrooms' effect on rohu hematology and gut microbiota. The main objective of this research was to evaluate the effect of dietary white button mushroom supplementation on hematological parameters like the red blood cells, white blood cells, hemoglobin, and other blood components of Labiorrhea to determine gut microbiota composition and bacterial colony counts to assess white button mushroom as natural immunostimulant in aquaculture. Its methodology includes feed preparation and experimental design. For the purpose of feed preparation, fresh mushrooms were taken and dried at the temperature range of 50 to 60 degrees centigrade, later ground into fine powder. The experimental design is divided into two experimental groups: control group fed with commercial feed and the treatment group fed with 2% white button mushroom powder mixed with feed. The experimental duration was 8 weeks. 20 fingerlings were kept in each group. Total 40 fingerlings of rohu were used in this experiment. Water quality parameters were maintained. Temperature was kept 24 plus minus 2 degrees centigrade. DO and pH level were also monitored regularly. Blood samples were collected from caudal vein of rohu, which later sent to the laboratory for the analysis of red blood cells, white blood cells, hemoglobin, and other blood component composition. Intestinal samples were collected from the gut of rohu that was cultured on 3 different medium nutrient agar, tryptin soy agar, eosine methylene blue to check the growth of bacterial colonies. Later, for bacterial identification, gram staining and biochemical tests were performed. In statistical analysis, data analyzed using t-test in MS Excel software, significance level was kept less than 0.05, result expressed as mean plus minus standard deviation. When the result for gut microbiota was analyzed, there is a magical increase in bacterial colonies in T1, which is known as experimental group, across all media. Here is the table of nutrient agar medium, where you can clearly see the CFU count of control group is significantly lower than the experimental group, and there is a much more elevation of CFU count in experimental tank. When bacterial species identification tests were performed, it was seen that in experimental group, the beneficial bacteria like Bacillus subtilis and Enterococcus faecalis were significantly increased, and the pathogenic bacteria like Aeromonas hydrophila and Staphylococcus aureus were significantly decreased. This showed the success of this experiment. White button mushroom promoted probiotics growth and suppressed pathogens. When the hematological parameter is analyzed, it was seen that the red blood cells and hemoglobin content of the blood was increased, that improved oxygen carrying capacity and indicate better metabolism. White blood cells composition was also increased, that indicate the enhanced immune response. Similarly, hematocrit, mean cell volume, and mean cell hemoglobin composition was also increased, that indicate better erythropoiesis and overall health of rohu fish. The t-test table is shown here. Here you can see red blood cells, mean and standard deviation, and the p-values. which clearly show the success of this experiment. In discussion, white button mushroom acted as prebiotics, promoting beneficial gut bacteria. High CFU count, improved nutrient absorption and disease resistance. Reduction in pathogens indicate healthier gut environment. The discussion of hematology and immunity, beta glucans and antioxidants in white button mushrooms stimulated erythropoiesis, red blood cells production, and leukocytosis, white blood cells increase. The result is clearly aligned with previous studies on mushroom-based feed in other fish. From this experiment, it is concluded that dietary white button mushrooms significantly improved gut microbiota diversity and beneficial bacteria, hematological parameters like red blood cells, white blood cells, hemoglobin, and hematocrit. White button mushroom is a sustainable natural immunostimulant for rahu aquaculture. And from this experiment, it is recommended that farmers can use white button mushroom in commercial aqua feeds as it is already a tested feed. For future studies, white button mushroom dose dependent effect should be analyzed and its molecular mechanism should be investigated, like how it affects on immune gene expression. Test against specific pathogens like disease challenge trials should be also checked.
So for our next presentation, I would like to invite Ms. Mahvish from University of Sialkot, and her topic of presentation is assessment of selected heavy metals, accumulation in feeders of scavenger bird species from district Sialkot. Kindly proceed with the presentation, Ms. Mahvish.
Assalamu alaikum. I am here to present my research work. First, here is my title, Assessment of Heavy Metal Accumulation in Feeders of Scavenger Bird Species from District Sialkot. My name is Mavish. I am from Zoology Department at University of Sialkot. Here is my table of content. First, I discuss about introduction, problem statement, objectives, material and method, statistical analysis, result, and here is graphical abstract of my title. Landfill and dumping sites. Here we collect here we select three species: crow, myna, kite. Then we collect feeders of these species. Next, digestion of sample, and at the end we for the metal detection used induced coupled plasma mass spectrometry, and at the end results are come. First of all, introduction. Landfills and dumping sites are major solid waste concentration sites attracting large assemblies of scavenger birds. Scavenger birds are those animals that consume dead things that have perished for reasons other than predation in the environment. At the highest trophic level, scavenger birds are especially vulnerable to the bioaccumulation of harmful substances in the environment. The atomic weight of heavy metals is greater than that of water molecular weight, and certain of them heavy metals, selenium, molybdenum, cobalt, and copper, play a part in metabolic processes. They will become harmful at high concentration. Heavy metals build up in the various animal body tissues after consumption, including bird feeders. Feeders are linked to bloodstream during growth, allowing metal to bind to protein molecule, making them useful for monitoring heavy metals. Feeders store heavy metals and high contamination level can disrupt physiological processes, threatening a bird's health, reproduction, and its survival. Choosing birds that react predictably and are sensitive to environmental changes is crucial for managing the pollution of heavy metal in the environment. For this reason, the current study uses feathers of kites, house crows, and common mynas to analyze metal pollution. These birds are associated and typically reside in areas where people live. These birds also consume fruit, small animals, rubbish from human homes, etc. Next, we further move to the next slide. Here it is problem statement. Heavy metal toxicity is a major environmental issue. It can affect the health of ecosystem. It is crucial to measure the level of heavy metals in animals. Wild birds are used as a bioindicator of metal exposure in the natural environment. Next, here is objective of my research work to evaluate metal accumulation in feathers of different scavenger bird species from district Sialkot to assess the variable heavy metal concentration in different habitat types. Next, we move material and methods. We choose study area where we collect sampling. We choose district Sialkot. In district Sialkot, have four tehsil Sialkot, tehsil Samrial, tehsil Pastur, tehsil Daska. In these four sites, we also select different subsites where we collect our samples of three species: crow, myna, and kite. Experimental overview. There are basically four steps: sample collection, digestion of sample, metal detection, and statistical analysis. Sample collection. The feeder samples of each bird were collected from birds surrounding the area of waste dumping and landfills in different tehsils of Sialkot. The feeder sample was kept in polythene bags and carried to the laboratory for further analysis. Experiment overview of my experimental design. I select district Sialkot, in which have four tehsils: Daska, Samrial, Pastur, Sialkot, and number of sample that I collect from each subsite is 36. Digestion of sample. Here are the steps of digestion of samples. First one is sample cleaning. Wash feeders sample 3 times with distilled water to remove external contamination. Then dry the sample at 80 degrees C for 2 hours in an oven. Sample preparation is a third point. Cut dried sample into small pieces and weight 0.5 gram. Transfer the sample into a flask, adding 1.0 ml nitric acid and 0.25 ml perchloric acid for the digestion. Heating the sample gradually from low to high temperature. Then cool the sample at room temperature and add 2 to 3 ml distilled water to finalize the volume for analysis. The digested sample is now ready for heavy metal analysis by using induced coupled plasma mass spectrometry. Next, I used statistical analysis, in which I used RStudio. Two kinds of tests used: ANOVA and PCA. Here are the results. This is the graph of average values of heavy metals in kites across selected sites of Sialkot. These 12 heavy metals show the different concentration across sites. Arsenic mean. In arsenic, there is a high concentration in site 3 and site 4. In cadmium, there is a high concentration in site 1. In cobalt, there is all negative values, so bar moved downward. In chromium, there the maximum concentration present in site 1. And here we and then they move the upper graph, the length show the high concentration and the small length short bars show the lowest concentration of heavy metals. Next is metal concentration analysis across sites in mina. Again, graph represent the maximum concentration of heavy metals and also tell the sites which site contain maximum amount of heavy metal and which site contain minimum amount of heavy metals. And here is a graph, average values of heavy metal in Crow across selected sites of Sialkot. And this is a PCA byplot of heavy metals in Kite. Here basically we take four sites of district Sialkot, and this graph shows different sites, site 1, site 2, site 3, site 4, and the arrows present in it show the concentration of metals that were present in it a large amount. Like the arrows, size of arrow also show the tendency of heavy metals. Site 1 show the concentration of molybdenum and copper, and site 2 show the concentration of selenium, cadmium, chromium, and nickel, and site 3 show cobalt and lead, manganese, and site 4 show the maximum concentration arsenic, zinc, and potassium. And here is again BCA by plot of heavy metals in Crow. More heavy metals present in site 1 and more heavy metals present in site 2. This means that site 1 and site 2 contain more toxicity levels. And next at the end we compare the three species having 12 heavy metals, and in this graph show more heavy metal concentration present in Crow. And there is another second one, heavy metal concentrations present in Mina, and the last one is Kite. Here is my references. Thank you for your attention. Hope you like it.
Thank you, Ms. Mehbish. Your findings on heavy metal contamination are crucial for understanding environmental pollution and its impacts on avian species. So for our next presentation, we have with us Tabinda Jabin from University of Sialkot, and her topic of presentation is Assessment of Ectoparasitic Infestations and Associated Risk Factors in Free-Range Backyard Chicken Gallus gallus of District Sialkot.
Assalamu alaikum. My name is Tavinda Javin. I'm a scholar, Department of Zoology, and from University of Salkot. My presentation of my and research topic is assessment of ectoparasitic infestation and associated risk factor in free-range backyard chicken, Gallus gallus from district Salkot. These are the table of content, and these are the graphical abstract from our presentation. The chicken and related poultry species are source of animal protein worldwide. In response to rising global demand, chicken meat production has increased annually from 60 million tons in 2000 to more than 100 million tons in recent years. The frequency, variety, and risk factor of ectoparasite infestation in backyard chicken are examined in this study. A random sample of 72 chicken from both healthy and sick chicken from four different sites: Sambrial, Daska, Pastur, and Sialkot. Ectoparasite were collected and species were identified through manual removal and stereo microscope. Identification was done using established morphological keys sourced by 1982 after cleaning and preservation procedure. Ectoparasite were observed in all birds but were inspected. The clinical burden of ectoparasite were higher in diseased birds as compared to the healthy ones. These results demonstrate a strong correlation between birds' ectovirus infestation and its overall health. The next is introduction. The poultry industry has dominated the world widely with the production of egg and meat. It also plays a role in the national economic and public health provided by providing animal protein. Poultry share in national demand to increase 5 to 30% by 2030. Meat production must increase by 235% and egg production by 8 to 28% to meet demand. The common ectoparasites include lice, mites, fleas, and ticks can quickly spread from one bird to another due to overcrowding and poor hygiene. Ectoparasite led to weight loss as 7 to 11 gram per bird, which resulted in decrease in the egg yield of about 66 eggs per bird every year. Free ranging poultry at a higher risk of infestation as compared to the captive poultry benefit from better management. And the next step is problem statement. Ectoparasite infestation in free range backyard chicken Gallus gator led to significant health and production losses. The objective to evaluate the impact of associated risk factors such as habitat, diet, feed, clinical burden of ectoparasite in backyard chicken. To investigate the ectoparasite devastating backyard chicken Gallus gator from district Sialkot. The study area of district Sialkot. And the next step is methodology. The step of methodology includes step sample collection of ectoparasite, examination of ectoparasite, identification of ectoparasite, and preservation of ectoparasite. The sample collection. There are about 72 chickens from four different sites: Pasrur, Samrial, Salkot, and Daska. And these sites from every site from healthy and diseased chicken collected. The first step is animal examination. Chicken, external body part, comb, eyelid, wattle, neck, feathers, legs were inspected using a hand lens, and the collecting of ectoparasite using a fine soap brush. Mite and ticks were collecting using a knife blade. The preservation of method transfer to acetic glycerin, 1% glycerin, acetic acid, and glycerin for preservation, and ectoparasite was stored in ethanol with label bottle, age, sex, parasite type, and health status. The treatment of ectoparasite was treated with potassium hydroxide to soften a clear specimen for better microscopic observation. And the slide preparation, mounted on glass slide, a small drop of acetic glycerin in 1% of acetic acid in glycerin, and identification of ectoparasite using a stereo microscope. The identification used by Walls and solved by morphological classification. These are the sphere microscope data used is to identify the larger ectoparasite. The associated risk factors include habitat, overcrowding, hygiene, and management system. There are four tests that are used in the t-test: diagnostic, indus, logistic, regression, and one-way ANOVA. And these are the used in one-way ANOVA from four different sites. The x-axis is number of octoparasites and y-axis represent the site. The orange bar represent the ill birds, and the healthy represent and the blue bar represent the healthy birds. These results show the ill birds in ectoparasite infestation are larger than the healthy birds, and the result is highly significant. And the next is comparison of ill and healthy birds from the two group, and and the and these results show that the health ill birds highly significant and the highly active infestation as compared to the healthy birds. And the next is a species abundance across site. T-test used in the x-axis number of active birds and y-axis is site. And there are six different species that identify in four different sites. And these are t-tests used in this research. And the next is used in diversity index. The taxa individual dominus, Simpson, Shinon, evenness, richness, Fisher, Alpha, Burger, Parker, and Coca. Domin number of species richness हर साइड में फोर स्पीशीज है नंबर टोटल इंडिविजुअल पॉपुलेशन साइड पर साइड डोमिनेंस species are highly increased that's why dominance increase dominance diversity measure higher more diverse subsite में name similar diversity Shannon measure species diversity considering both richness evenness value 1.34 to 1.3 are similar across site evenness average species distribute equally 0.95 to 0.9 and vary even. Alternative to Shannon, when sampling is not random, similar values show uniformly. Richness relative to sample size very close, very uniform richness. Another richness, all 0.51 same richness. Equal distribution of individual among species 0.97 to 0.9 almost equal. Diversity parameters सभी side same 0.63 and similar. Show dominance of the most abundant species higher value more dominance side for 0.357 highest dominance. Estimated two species richness सभी side में 4 कोई hidden species expected नहीं है. And these are the logistic regression model of associated risk factor of hygiene, logistic regression model of associated risk factor of overcrowding, and logistic regression of the associated risk factor of habitat, and the logistic regression model of associated risk factor of management system. The last one is conclusion. These are the there are six different species identified in this research. Active virus infection, bacterial highly exotic and district alcohol. Disease birds show a higher burden of active virus compared to the healthy one. Major risk factors include hygiene, overcrowding, weak management system, and habitat condition. Active virus and negative effect the health, productivity, and farmer economy. Highlight the need of better biosecurity, farmer awareness, and preventing measure. And these are the reference. Thank you.
So, for our next presentation, we have with us Amna Nadeem from UF, and her topic of presentation is toxicological effects of copper sulfate on histopathology of Oreochromis niloticus. Ma'am, please kindly proceed with the presentation.
Assalamu alaikum everyone. I am Amna Nadeem, a postgraduate student from Department of Zoology, Wildlife and Fisheries at the University of Agriculture, Faisalabad, Pakistan. My presentation focuses on the toxicological effect of copper sulfate on histopathology of Oleochromis niloticus, which is the Nile tilapia, a significant species in freshwater aquaculture globally. Communities around the world depend heavily on freshwater and marine fish farming. These practices are vital for food security, nutrition, revenue generation, and livelihoods. However, the health of these aquatic ecosystems is threatened by water pollution. Pollutants can impair the immune systems of fish and other aquatic organisms, making them more susceptible to diseases. With the increasing pace of urbanization and Industrialization. There is a notable rise in the levels of organic compounds and heavy metals in water bodies. This situation underscores the need to study the impact of specific pollutants like copper sulfate on aquatic life. In my research, I will be discussing the toxicological effects of copper sulfate on histopathology of Nile tilapia. Understanding these effects is crucial for assessing the risk associated with copper sulfate in aquatic environments and for developing strategies to mitigate potential harm to fish populations and the broader ecosystems. Heavy metals are metallic elements with high atomic weights and densities. The discharge of wastewater from homes, businesses, and the use of anti-foiling paints are major cause of copper presence in aquatic environments. Fish exposed to heavy metals may suffer from a number of harmful consequences. For all living things to be healthy, copper is necessary micronutrient that is involved in many different cellular processes. Copper sulfate is used to control fish endoparasites, external bacteria, and fungi for therapeutic purposes. It is also widely used in industries like construction, farming, electronics, and as an antifungal agent in poultry. Exposure to copper sulfate can lead to decreased body mass, elevated oxidative stress, poor feed intake, and biochemical and also histopathological changes. The objectives of my study were to examine the toxicological impacts of copper sulfate on Oreochromis niloticus and to investigate various histopathological alterations in different organs like the liver, gills, muscles, and heart of Oreochromis niloticus after exposure to copper sulfate. The study followed these steps for histopathological analysis. First of all, tissue preparation. Preparation of tissues from Oreochromis niloticus for histopathological examination. Second one is fixation, fixing the tissues to preserve their structure. Third one is dehydration and clearing, dehydrating tissues and clearing them for further processing. Fourth step is impregnation, impregnating tissues for embedding. Fifth one is embedding. Embedding tissues in a medium for sectioning. Section cutting. Cutting sections of the embedded tissues. Seventh and the last, mounting and staining. Mounting sections on slides and staining them for further microscopic examination. I am presenting findings from a histopathological study on the effects of certain stressors on Oreochromis niloticus like copper sulfate. First of all, in the gills, severe hyperplasia and hypertrophy of epithelial cells, along with extensive inflammation, congestion, hemorrhage, widespread necrosis, and degeneration of gill tissues were observed. In the liver, we observed severe hepatocyte vacuolation and degeneration. There was extensive inflammation, congestion, and hemorrhage. The liver architecture was disrupted, including loss of sinusoidal spaces and the formation of fibrotic tissues. Moving to the heart, we found severe myocardial degeneration and necrosis. Extensive inflammation and hemorrhage were noted. There was widespread apoptosis of cardiac and myocytes. Disruption of cardiac architecture was widely observed. Lastly, in the muscle tissues, we saw widespread apoptosis of muscle fibers. Muscle architecture was disrupted, including loss of muscle fiber organization. These findings highlight significant histopathological changes in Oreochromis niloticus tissues. last, conclude. Animal histopathological alterations, particularly in fish, are strong makers for prior exposure to aquatic environmental stresses. Fish may experience stress from toxic copper, which can impair a number of biological processes and result in certain histopathological changes. Fish tissues may exhibit macroscopic abnormalities and histopathological alterations due to metal exposure. Thank you for your attention.
Thank you for your clear and detailed presentation. Your work highlights significant toxic effects of copper sulfate on effective organisms. So for our sixth presentation, we have with us Muntazir Zainab from University of Sialkot, and her topic of presentation is spatiotemporal distribution and population abundance of black kites, Malvis migrans in district Sialkot. Please begin your presentation, Ms. Muntazir.
Assalamu alaikum. My name is Muntazir Zainab from University of Sialkot and Department of Zoology. I am going to present my topic Spatial Temporal Distribution and Population Abundance of Black Kites, Milvus migrans in District Sialkot at the 8th International Conference on Applied Zoology 2025. Introduction. The family Streptridae contains 10 common groups of birds that prey on other small birds such as kites, hawks, European vulture, snake eagles, dusky hawk, etc. The raptors Milvus migrans, commonly referred to as the black kite and belonging to the Streptridae family, is locally recognized as the Kalu cheel. Kites emerge as significant bioindicators of ecosystem well-being. Kites exhibit fundamentally similar feeding behavior, leading to the observation of interspecific kleptoparasitism at a common feeding site. Common kite Milvus migrans often gather in large communal roosts and breeding colonies. Large aggregation also occur where food is abundant. Black kite is closely associated with the human settlement. Problem statement. Population distributional studies provide the critical insights for the biodiversity management and conservation efforts. The black-eyed milvus migrans population is affected by urbanization, habitat changes, and seasonal variation. So, this study will help to uncover population dynamics of black kite in district Sialkot. Research objectives: to examine the spatial temporal distribution pattern of kite in district Sialkot and to check the effect of climate variable like temperature, relative humidity of abundance of kites. Methodology: materials and method, study area, survey method and statistical analysis. Study area. This slide shows the study area of my research, which is district Sialkot in the Punjab province of Pakistan. Sialkot was selected because it has a mix of urban and rural habitats that are important for black kites, providing both roosting and foraging sites. This study covered 4 tehsils in the district Sialkot, and black kites are commonly found where the ecological factors like food and water availability vary. In the next slide, survey method. There are three survey methods: line transect, point count, and roost count method. In line transect method, a total 8 transects were taken in each site in each tehsil of Sialkot. In the second point count method was used where birds are counted from fixed points in the field to assess population density. And in the last, roost count method is used where the birds returning to the communal roost were counted. Statistical analysis. The R Studio is the software is used during the research, and tools one-way ANOVA, Tucky post hoc test and regression were used. In the figure 1, this data reflects significant number of birds at the urban areas across all 4 sites. Highest number of kites are present at site 1 due to the availability of food, and the lowest is present in the site 2. You can clearly see from the figure that all the urban sites has the more population than the rural site. In the figure 2, during the study period from November to the May, the population of black kite gradually increased across all four sites. The number were lowest in the winter from November to December, and then the lowest in the February again. The number is started increasing from March to May. This is because the breeding season of the kites is summer season. In the figure 3, a closer look at the site shows that the site 1 has more population than the other four sites. Site 2 were recorded as the lowest, and it shows the comparison in between the four sites. You can see that the site 3 and 4 had moderate population with no significant differences between them. This indicates that factors like food availability and roosting site play a key role in determining kite number in each location. In the last figure, you can see the spatial temporal distribution of the black kites in the map, where the number of species in the rural and urban site is lower in the November, December and January, and again in the March to May the place show the highest number. Some kites may migrate to warmer region, further contributed decline in some regions, and some region has the highest number. In the both figure, linear regression is shown in between the temperature and humidity. Temperature showed a positive correlation with black kites, and humidity shows the contrast relation with the black kites. Conclusion: major threat to raptors include deforestation, habitat fragmentation, which significantly limit their population. In four tehsils of district Sialkot, there is fluctuation has been seen during different seasons. The highest population of black kites in Tehsil Sialkot is due to greater availability of food resources, and in contrast, the lowest population is Samadial may result from less favorable environmental condition, reduced food supply, and fewer nesting sites due to less urbanization of agriculture activity. The high activity of black kites observed at dump areas likely due to the abundance of availability of food such as organic waste and carrion. In the end, I would like to express my deepest gratitude to all my colleagues and my teachers who contributed to the completion of my thesis. A warm thanks to my supervisor, Dr. Sabah Manzoor, for her support and guidance and encouragement. And I am really grateful to the faculty head, Dr. Roma Adalat, for providing resources and assistance. At the end, I would like to say thanks to my family for their unwavering support and understanding, which motivated me to the preserve. This is the end. Thank you so much.
Thank you for such valuable insights on population dynamics and habitat distribution of black kites and fascinating part of urban ecology. So for our next presentation, we have with us Tayyaba Israr from University of Sialkot, and her topic of presentation is biomonitoring of selected heavy metals in grey partridge species from Hadmarala, Sialkot. Please go ahead, Ms. Taiba.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. Myself Taiba Shafiq, and it's my pleasure that I'm here to represent my research work in the 8th International Conference on Zoology in 2025. I'm from University of Sialkot in the Department of Zoology. Here are the contents of my today's presentation. The first one is introduction, materials and methods, results, conclusion, acknowledgement, and then references. Introduction, as we know that all ecosystems naturally include metals, numerous inner states of organisms are associated with either an excess or a deficit of essential metals. Metals can contaminate both terrestrial and aquatic ecosystems. Metals including like arsenic, cadmium, and copper have been linked to genotoxicity in birds that can decrease their birth rate. In this study, pathogens were used that belongs to family Fassinidae and order Galeriformes. The problem statement of this study is that Agricultural and industrial waste contain different types of toxic chemicals and heavy metals. These heavy metals become the part of food of different animal species, including parasites. So it is important to investigate these metal concentrations in parasite species. The significance of the study is to create awareness among the public about this alarming situation that may cause decline in population of grey parasites in near future. This study will be helpful for the management, protection, and improvement of health of parasites and the native environment. In study area, Sialkot were choose. The Hadmrala is a picnic spot and wildlife reserve located in the Sialkot in the Punjab province of Pakistan. Three sites were selected from the Hadmrala. The first one is Marala, Raviling Canal, Upper Chenab, and Marala Headworks. Then the selected species that is strategies were captured from three sites. Then three samples were collected from each site. In sample collection, the strategies were dissected by using dissection plates and collect the organs, then transfer these organs in lab for further analysis. In analysis, the first one is digestion of feeders. In feeders digestion, wash the feeders with table water and acetone for remove external contamination. After this, let them air dry for 2 to 3 hours. After the feeders were drying, add a mixture of acids, hydrochloric acid and hydronitric acid. Then the digestion of the feeders will start, and after the digestion, dilute the mixture with 25 milliliters of deionized water. After the dilution, the filtration will take place by using Wardman filter paper and send these samples for further analysis of metals by using ICP-OES. In digestion of organs, there is a minor difference. Let them over drying the organs for 24 hours. After the drying, their weight was measured by using computerized dry weight by using computerized measuring balance, and then add a solution of mixtures of acids, hydronitric acid and hydrochloric acid. After this, the digestion of the organs will take place, and after the digestion, the samples were dilute by using hydrogen peroxide, and after the filtration will take place using Wardman filter paper, and then after then dilute the mixture by using 0.1 milliliter, and then send these samples for further analysis of metals by using ICP-OES. In the result, this bar graph shows the metal concentration in uh, organs and size of particles. The arsenic is a metal that shows the significant differences between across the size and across the organs, and the rest of the graphs also show the different metal concentrations in three sites and three organs that vary significantly. All 11 metals show statistically significant differences across the three sites. Their p-value is less than 0.05, and the metal concentrations vary always significantly across organs, that is liver, kidney, and feathers. Also show the significant difference, and the highest concentration overall is uh, shown in liver, up by kidneys and feathers. In conclusion, the highest concentrations of heavy metals were observed in the liver, followed by the kidneys and feathers. And the observed differences in heavy metal concentrations across sites suggest varying levels of environmental contamination. Iron was found in higher concentrations in the liver at site 3, and copper consistently shows very low concentrations across all tissue types and sites. Acknowledgement. First and foremost, thanks to Allah Almighty for being my strength and guide me through this journey. The special thanks are offered to Dr. Sabah Manzoor for her exceptional guidance, mentorship, and expertise through this research journey. Her patience, encouragement.
Thank you, Ms. Tayyaba, for this significant contribution on biomonitoring and ecological risk assessment in local wildlife species. So, for our next presentation, we have with us Ms. Khatiba Saeed from University of Sialkot, and her topic of presentation is effect of butyrate-based dietary supplements as growth and immune boosters in desi chicken Gallus gallus.
Let's start with the name of Allah. Bismillahirrahmanirrahim. My name is Hatiba Said and I am affiliated from University of Sialkot. Here is the topic of my abstract: effect of butyrate based dietary supplement as immunity and growth booster in desi chicken Gallus gallus. Here it is the table of contents of my presentation, and there it is the graphical abstract of my presentation in which I show the effect of butyrate. First I taken a desi chicken and then I applied a supplement which is a phosphorus SR130. I designed 4 experiment groups and to show how the effect of butyrate supplements on the chickens, I taken a blood sample from each chickens. Then I analysis the data analysis of the blood group, and here then I show the how it is the effect on growth, on immune system, and on biochemical parameters. After data analysis, I making the graph of these parameters. And the introduction of my presentation is the poultry, meat and egg production have shown a considerable increase worldwide. The increase in the size of the poultry industry has been faster than other food producing animal industries. Under modern system of poultry production, birds are inevitably exposed to considerable stress during their productive lifetime. The general trend in the poultry industry is to find safety for the bird, whether in the feed or the environment conditions. In Pakistan, poultry sector contributes to enhance the economy of country by generating employment for about 1.5 million people, as well as in meat production centers contributing 90% of the total meat. To overcome the bird stress and improve their growth, butyric acid is used. Several studies demonstrated that supplementation of organic acid, which include the butyric acid, formic acid, and acetic acid, especially butyric acid, to broiler diets increased growth performance, reduced diseases, and management problems. The objectives is first objective is that to evaluate the effect of butyric acid on growth performance of callus, and second objective is to investigate the effect of butyric acid on biochemical profile and hematology of callus. Problem statement of this research is the digestive and immune system problem, which are poor digestion, nutritional deficiencies, or infection in chicken can significantly affect their growth, productivity, and overall health. The materials and method contain the following steps. First, we selected the species, then applied the dietary supplement, and then design an experiment. Selection of species contained 36 unsexed male and female desi chicken that appear to be in a good health, woodhoused in sanitary circumstances. Study consisting of the 4 experiment groups, 3 replicates per group of 4 chickens in each. Dietary supplement: butyric acid supplement used provided in the form of commercial powder product Prosperous SR130 Perstop is the brand. Supplements mixed with the basal feed basal diet, and the components of the basal feed diet is the corn, choline, mild, MCP, L-lysine, phytase, rice, milk, metabolic energy, ether extract, crude fiber, and methionines. These are the basically basal diets components, and I used the Olympic feed basal diet. The experiment design contains 4 groups. First one is the T0, which is the control group, T1, T2, and T3. T0 is the feed basal diet without butyric acid to each groups, to male and to female. And T1 is the 0.5 gram butyric acid per kg basal diet during the first period from 1 to 21 days, then basal diet until day 42. T2 feed 0.5 gram butyric acid per kg basal diet during the whole period from 1 to 42 days, and T3 feed basal diet from 1 to 21 days and then feed 0.5 gram butyric acid per kg basal diet during the second period from 22 to 42 days. These are the basically my protocol of my experiment. The parameters which are found out in this research is the growth, performance parameters, blood hematology and biochemistry and histological techniques. Here I shown the blood collection sites where I taken blood from the chickens. Three main sites are basically used from which we collect the blood from the chicken: brachial wing vein, jugular vein and medial metatarsal vein. Mainly the vein which is used from which blood is collected is the brachial wing vein. Here is the protocol of my blood hematology and the biochemistry. First I take a chick, three chicks from each group, and then I collected the blood. After collecting the blood, I place it in the EDT tube that not blood coagulated, and then mixing the tube to separate out the serum and the blood. For this I used mainly centrifugation techniques which separates the blood to the serums, and the serum is used for the biochemical assays and the blood is used for the hematological analysis. After hematological analysis we making different crops. Here is the statistical analysis which I supplies in this research. I used the R software 4.4 variants in which the test we applied is the one-way ANOVA post-Hawking test. And then if my test is significant, then I applied the Deacon test for lettering. Here is the results of my research. First I find out the effect of butyric acid on growth, performance and feed efficiency in desi chickens. These are the parameters which I find out in my research: live body weight gain, FC mean feed consumption and feed consumption ratio and mortality rate. In first day my live body weight is increased in the T group, and the p-value show the significant differences among the groups, and in at day 21 and 42 days T show the highest body weight gain. And after the live body weight I find out the body weight gain, FC and FCR ratio. Body weight gain also increase in T2 group, and the p-value show the significant differences among the groups. FC mean the feed consumptions, that which group taking the feed more than as compared to increasing their weight. So T0 mean T control group show the highest feed consumptions among all the groups, and the FCR is also increase in the T0 as compared to the other groups. These lettering show the which groups have increase in number as compared to the other groups, and the mortality rate also show the non-significant differences. Here is the blood hematological analysis. I shown the different graphs. First graph is shown the comparison of PCV value among the groups. Second graph shown the comparison of hemoglobin, and third graph shown the comparison of red blood cells across the treatments. Here it is the result that the PCV and hemoglobin and RBCs improved significantly across the groups, and T2 group showed the highest PCV, hemoglobin and RBC values. PCV, hemoglobin and RBC follow the trend. T2 is the highest, then T3, then T1, and then T0, indicate enhanced erythropoiesis and oxygen carrying capacities. Here it is the blood biochemistry. Treatment significantly influenced glucose, cholesterol, and the triglyceride, which is shown in the graph. T2 in T2 was the highest effective in lowering cholesterol compared to the control group, and T3 reduced triglyceride levels among all the groups, and T1 and T2 produced a slight reduction in glucose, while T3 maintained the highest value. Then my kidney profile show that keratin is the highly significant lowest in T3 better clearance, and urea is a highly significant reduction in T2 and T3. Uric acid is significantly lowest in T2, indicated improved metabolism, and T2 and T3 improved the kidney function as compared to the other group T1 and T0. And the liver profile, the comparison between ALT and AST levels, in which shown that significant differences observed among the groups. T2 shows the lowest ALT and AST, indicated minimal liver stress and better protection. T1 also lowered the enzyme level compared to the control, suggested moderate improvement, and T3 had low ALT but higher AST pointed towards mild liver stress. And the conclusion of my presentation is the study concludes that continuous supplementation with butyric acid significantly improves growth performances and immune response in the desi chicken. That's all from my side. These are the references of my presentations. Thank you so much. Allah Hafiz.
Thank you, Ms. Khatiba Said. Your findings add great value to poultry health and nutrition, especially in enhancing immunity through dietary supplementation. For our next presentation, we have with us Faiza Khanum from University of Veterinary and Animal Sciences, and her topic of presentation is dietary supplementation of Hydrilla verticillata extract improved growth, physiological responses, disease resistance of Labiorhita rohu. Please begin your presentation, Ms. Faiza.
Assalamualaikum everyone. My name is Faiza Khanam and my topic of presentation is dietary supplementation of Hydrilla verticillata extract improves growth, physiological responses and disease resistance of Labiorhita. First of all, I will tell you about my abstract. The present study evaluated the influence of Hydrilla verticillata extract rich in phytol on growth performance, antioxidant stress, immune response and disease resistance in Labiorhita after challenge with Aromonas hydrophila. Fish with an average initial weight of 11.5 were fed diets incorporated with 0.75, 150, 300 and 600 mg per kg of Hydrilla verticillata extract for 60 days. For feeding trial, 30 fish were randomly distributed to each of the 5 aquarium in triplicate. The 300 mg per kg group was significantly best in increasing weight gain, SGR, FCR and PER. The proximate analysis of whole body showed non-significant differences between all groups compared to the control group. Antioxidant enzyme activities were significantly highest and the lipid peroxidation was minimum at 300 mg per kg of Hydrilla verticillata extract. Fish fed with 300 mg per kg showed the highest level of serum alkaline phosphatase, ALT, AST, glucose, and cortisol were lowest as compared to the control group, reflecting less stress and improved liver function. Moreover, the level of serum lysozyme and immunoglobulin were recorded significantly highest in group fed with 300 mg per kg of Hydrilla verticillata extract. This fish group also showed the least mortality after challenge with Aeromonas hydrophila, indicated increased resistance to disease. The broken line regression showed that 252.46 mg per kg addition of Hydrilla verticillata extract in diet was optimum to improve growth, physiological responses, and immunity of Labiorhita against Aeromonas hydrophila. Now we will discuss about the introduction. First of all, I will tell you that the aquaculture fish production increased significantly day by day, and last few decades it was the highest. The enormous and rapid production increased to contagious diseases caused by the opportunistic pathogens. To control these diseases, we mostly use the antibiotics, but now immunostimulant are used is a chemical drug or action that enhance the defense mechanism or immune response, thus rendering the animal more resistant to disease. Herbs are an interesting alternative of vaccines because they are inexpensive, renewable, locally available, user-friendly, and can be easily prepared. So Hydrilla verticillata is a herb which can be used, and it is enriched with nutrients and chemical constitutions, amino acid, antioxidant agents, and among all compounds, phytol is present in large amount and has tremendous antinociceptive and antioxidant activities. Now we go to the objectives of this thesis. To determine the effect of Hydrilla verticillata extract on growth performance, proximate composition, and hematological parameters of Labiorhita. To assess the impact of Hydrilla verticillata extract on antioxidant enzyme activities and stress response in Labiorhita. To evaluate the effect of Hydrilla verticillata extract on serum biochemistry and immune status of Labiorhita. Now this is the content of the materials and methodology of this topic. So the methodology is the research work was carried out at Fish Seed Trading Unit, Department of Fisheries and Aquaculture, UAS, Ravi Campus, C Block, Pataki. The experimental species was Rahu Labiorhita fingerling was used. First of all, the extraction of Hydrilla verticillata extract was collected from University of Veterinary and Animal Sciences, then washed with distilled water and shade dried, then sample was grind into powder. Then we use the Soxhlet apparatus for extraction with 95% alcohol for 5 hours. And then the extraction was collected. This is the composition of the vessel diet. So the formulation of diet was simple. Dry ingredients were used, then ground and screened in grinder, add minerals, mixer, fish oil, vitamin premix, then place in meat mincer, make dough, add water, pellets formed, and then dried and then stored in vacuum bags. So the experimental layout was the vessel diet, then added as the supplementation is a Hydrilla verticillata extract, and this was used at the level of the 0, 75, 150, 300 and 600 mg per kg, and the diets were D1, D2, D3, D4 and D5. So the experimental design was completely randomized and 20 fish were stocked randomly in each of the 15 aquarium. Every experimental diet was fed to the triplicate aquarium. First, the fish husbandry adjust all the pH, temperature and the DO, and the trial was continued for the 90 days. And then fish harvesting at the end of the experiment, starved the fish for 24 hours, anesthetized the fish, whole body, blood, liver, serum samples were collected. And then the analysis of the growth performance,
Thank you for comprehensive research, Ms. Faiza. Your studies on hydrilla extract provides a promising natural alternative for promoting fish health and disease resistance. So this brings us end of highly engaging and informative session. I extend my heartfelt thanks to all our presenters and audience for active participation and keen interest. I hope today's session will inspire further research collaborations and innovations in field of applied zoology. Thank you all once again. Stay connected and enjoy the remaining session of conference. Allah Hafiz and have a wonderful day ahead.